Imagine placing your hand directly onto a burning hot stove.
Right.
But instead of immediately pulling away in agony, you just stand there feeling perfectly fine.
Which sounds completely impossible.
Exactly. And not because you took a massive dose of painkillers or, because your nerves are damaged. You feel fine because your brain simply manufactured its own painkiller out of thin air based entirely on the belief that the stove was cool.
It sounds like a parlor trick, honestly, but it's actually one of the most robust and totally misunderstood phenomena in human biology.
Yeah.
For the longest time, the medical community treated this kind of thing, the placebo effect, as if it was just, in your head.
Just a reporting bias, right?
Yeah.
Like you just want to please the doctor, so you say you feel better.
Exactly. They viewed it as a statistical nuisance, an illusion.
Well, today we are taking a deep dive into a 2026 paper from the journal Neuron by a team led by Lavrisi and colleagues that completely dismantles that old way of thinking.
It really does. It's groundbreaking.
They've reverse translated a human phenomenon into a mouse model to map the exact physical brain circuits of the placebo effect.
Yeah, the actual hardware.
Right. We're going to see how the highest level thinking centers of your brain literally send chemical commands down your spine to shut off pain. It is a physical highway, and researchers can now flip it on and off like a light switch. Okay, let's unpack this.
So to understand how they mapped this highway, we have to look at how they built the foundation of the experiment. Because, you can't just hand a mouse a sugar pill, pat it on the head, and say, trust me, you'll feel great.
Right. Mice don't really speak English.
Exactly. You have to construct the expectation of relief from scratch. Because without expectation, a placebo doesn't exist.
So they used a contextual conditioning protocol.
They did. They took a group of mice, injected them with actual morphine, and then placed them in a very specific environment. Let's just call it the treatment room. Inside this room, the mice were put on a hot plate. Now, it wasn't hot enough to cause tissue damage, but it was uncomfortable enough that a normal mouse would, jump or lick its paws to cool down.
But these mice had morphine in their system.
Right, so they were perfectly comfortable. They could sit on that hot plate without any distress at all.
And they repeated this over several days, right?
Yeah, over and over. What they were doing was teaching the mouse's brain a really vital association. Basically, when I am placed in this specific room, I am protected from the heat. They were wiring the environmental context to profound pain relief.
And then comes the test.
The real test, yeah.
They put the mice back in that exact same room on the exact same hot plate. But this time, they swapped the morphine for a completely inert saline injection. Just salt water.
Which has absolutely zero chemical ability to stop pain.
And yet the mice just sat there, perfectly comfortable, withstanding the heat exactly like they did when they were floating on actual morphine.
Their brains literally manufactured a pain-blocking shield out of pure expectation.
But, a skeptic might look at that and say, well, maybe the mice just associate that room with the euphoric high of morphine.
Right, the good vibes theory.
Yeah, exactly. If I have a great memory of the restaurant, walking in makes me happy. Wouldn't the mice just be experiencing a lingering good vibe that distracts them from the heat?
That is a really crucial distinction, and the researchers tested it perfectly. They set up an unpaired control group.
Okay, how did that work?
They put these mice in the exact same room and gave them the exact same morphine. But they never turned on the hot plate.
Oh, I see.
So these mice learned that the room was a great place to get a pleasant drug, but they never once associated the room with relief from a painful threat.
And when they finally tested those unpaired mice on the hot plate with just saline,
they felt the full force of the heat. They jumped. They licked their paws immediately. There was zero placebo effect.
Wow.
This proves that the placebo isn't just a generalized memory of feeling good. The brain has to specifically link the environment to the alteration of a painful experience.
It's a predictive coding mechanism.
Exactly.
I also read there was an interesting demographic nuance here too, right? With male and female mice.
Yeah, that was fascinating. In these healthy, pain-naive mice, they found that the placebo effect was actually more robust in the male mice than the female mice.
Which is really interesting because when we look at human clinical trials, especially for chronic pain conditions, we sometimes see stronger placebo responses in women.
It just highlights how incredibly complex this neurobiology is, right? A healthy nervous system reacting to acute temporary pain might use these predictive circuits differently than a system rewired by chronic injury.
Right. But regardless of sex, the fundamental hardware exists.
It does. The mice proved the phenomenon is real.
So let's do a quick subtopic recap for you listening. Bullet 1. Placebo analgesia requires learning to predict pain relief in a specific context.
Yep.
And bullet 2. Morphine without the pain stimulus doesn't build the placebo expectation. It has to be paired.
Exactly right.
So I have a question for you listening. Think about your own life. Have you ever felt a headache start to fade the second you swallowed a pill before it could even digest?
Oh, I know I have.
Right. That's your predictive circuitry firing up. Your brain is an anticipation machine.
So now that we know it works, the next monumental task for the researchers was finding exactly where in the brain this magic trick was happening.
And their search took them deep into the ancient primal part of the brainstem, to a region called the ventrolateral periaqueductal gray.
Yeah, that is a mouthful. Let's just call it the vlPAG.
Good call. So, if your nervous system is a massive audio soundboard, the vlPAG is the master volume knob for physical pain.
That's a highly accurate way to visualize it. The vlPAG sits up in the brainstem and sends nerve fibers down into the spinal cord,
Specifically interacting with an area called the rostroventromedial medulla, or RVM.
Right. And when the vlPAG gets the right signal, it tells the spinal cord to literally close the gates. It mutes the pain signals traveling up from your body before they even reach your conscious awareness.
But the researchers didn't just assume the vlPAG was responsible for the placebo effect, right? They had to prove it.
And they did it using some absolutely stunning genetic engineering because, it's not enough to just know the general brain region. They wanted to isolate the exact specific neurons responsible for the expectation of relief.
So how do you even do that?
They use a genetic system called TRAP2. Think of it like a microscopic high-tech highlighter pen.
A highlighter pen.
Yeah. It doesn't just broadly highlight the whole brain. It is engineered to permanently tag only the specific neurons that are actively firing during a very specific window of time.
Okay, so right at the exact moment, the mice were sitting on the hot plate with saline, successfully experiencing the placebo effect.
Exactly. The scientists activated this genetic highlighter. It swept through the vlPAG and tagged only the specific cells that were driving that pain relief.
That is wild, but tagging them is only step 1, right?
Right. Step 2 is manipulating them. Once those placebo neurons were highlighted, the researchers used a technique called chemogenetics, specifically a system called
DREDIes, which is basically splicing a remote control kill switch into those specific highlighted cells.
Exactly. This switch sits completely dormant on the neuron, doing absolutely nothing until the scientists introduce a very specific, artificially designed chemical called CNO.
So picture this. You have these mice, they're put back on the hot plate, their expectation kicks in, and the placebo effect should be protecting them from the heat.
Right.
But then the researchers administer that designer chemical, CNO.
Right.
It washes through the brain, finds the remote control switches on those highlighted vlPAG neurons, and instantly shuts them off.
And the result was immediate. The placebo effect vanished entirely.
Oh, wow.
The mice suddenly felt the full, uncomfortable heat of the hot plate. By turning off just that specific handful of tagged neurons in the brainstem, they completely deleted the expectation of relief.
The volume knob got stuck on loud.
Precise.
It's just incredible to think about expectation as a physical object you can just switch off.
It really is.
Let's do another quick recap. Bullet 1. The vlPAG to RVM pathway is the physical volume knob for descending pain control.
Correct.
And bullet 2. Advanced genetic tools allowed researchers to turn off only the specific neurons involved in the placebo memory.
Which killed the effect.
Right. So, for you listening, if you could consciously control your vlPAG volume knob, what everyday annoyances would you tune out?
Oh man, I can think of a few.
Same. But this creates a huge logical puzzle, doesn't it? The vlPAG is down in the brainstem. It's a relay station. It's middle management.
Right.
In our experiment with the saline, there is no actual morphine in the blood to activate the brainstem. So if the vlPAG is just the volume knob, who is reaching down and turning it?
That was the next big question. They needed to find the CEO giving the orders. And to trace the communication lines backwards from the brainstem, they used a modified rabies virus.
Like actual rabies?
A safe pseudotyped version, yeah. But they used it because the rabies virus in the wild is terrifying precisely because of how it moves.
Right, it infects a nerve and travels backward.
Exactly. It jumps across the synapses, climbing up the nervous system toward the brain. The researchers hijacked that exact mechanism.
Oxford, really?
They injected this modified rabies into the vlPAG and let it act as a biological wire tracer. It jumped backward, lighting up every area of the brain that was sending signals down to that volume knob.
And they found a dense web of connections coming from high up in the cortex, the advanced thinking part of the brain.
Specifically, they found 3 major suspects: the medial prefrontal cortex, the anterior cingulate cortex, and the anterior insula.
Let's call them the MPFC, ACC, and AI. These are regions that handle complex executive function, expectation, emotion, and conflict monitoring.
They are the definition of top-level management.
So the researchers went back to their chemogenetic remote controls. But this time, instead of silencing the brainstem itself, they selectively silenced the specific communication pathways traveling from these cortical areas down to the vlPAG.
And this is where the paper fundamentally changes how we view pain. When they silence the pathway from the medial prefrontal cortex down to the vlPAG, the placebo effect was completely blocked.
The CEO was gagged.
Exactly. The brainstem never got the order to turn down the volume, and the mouse felt the pain.
Here's where it gets really interesting. When they silenced that exact same top-down pathway, but gave the mouse actual morphine, the morphine still worked perfectly.
Which is a crucial distinction. It perfectly illustrates the difference in causality. Real opioid drugs work from the bottom up.
Right. Morphine enters the bloodstream, floods the brain, and directly binds to the volume knob in the brainstem. It doesn't need permission from the thinking brain.
But a placebo is inherently top-down. You absolutely require your advanced cognitive cortex to actively process the context, generate the expectation, and forcefully command the primal brainstem to alter your physical reality.
It is the literal biological manifestation of mind over matter. If your prefrontal cortex can't communicate with your brainstem, your expectations are entirely powerless.
And we should mention that 3rd suspect they found, the anterior insula, or AI.
Right. What happened there?
That provided a really elegant contrast. The anterior insula is heavily involved in evaluating the emotional unpleasantness of pain. It tells you how bad something feels.
Okay.
But when they silenced the pathway from the insula to the vlPAG, the placebo effect still worked.
Oh, so the insula is like the HR department. It observes the situation, it takes notes on how unpleasant the heat is, but it doesn't actually have the authority to make operational changes.
That's a great analogy. The medial prefrontal cortex and ACC are the true CEOs making the call to shut down the pain.
Let's do another quick recap. Bullet 1. The MPFC and ACC act as the CEOs initiating the placebo effect. Bullet 2. Top-down cortical input is uniquely required for placebos, but not for opioid drugs.
Yep, completely different pathways.
So, question for you listening. Knowing your prefrontal cortex is actively modulating your physical sensations, does this change how you view mind over matter?
It definitely shifts it from philosophy to biology.
It really does. But, command from the CEO is just an electrical impulse traveling down a nerve. Once that electrical signal hits the vlPAG, how does it physically twist the volume knob?
Right. What's the actual chemical messenger that executes the order?
The software to the hardware. The answer is endogenous opioids.
Yes, the brain's naturally manufactured painkillers, things like endorphins and enkephalins.
And to prove that these were the specific keys unlocking the placebo effect, Lavrisi's team utilized a technology called fiber photometry, specifically a D-light sensor.
Right. The D-light sensor is a genetically encoded fluorescent molecule that they introduced into the vlPAG.
And what does it do?
It is literally designed to physically glow green whenever it comes into contact with the brain's natural opioids.
That's like sci-fi.
It is. By threading a microscopic optic fiber into the brain, the scientists could sit in a dark room and literally watch the brainstem light up in real time as the chemicals were released.
And the timing of what they saw is just mind-bending. When they placed the conditioned mouse on the hot plate, the brain didn't just slowly leak opioids over time.
No, it was instantaneous. The D-light sensor showed a massive, sharp spike in opioid release in the vlPAG precisely at the millisecond the painful heat stimulus was applied.
The brain recognizes the context of the room, anticipates the incoming threat, and preemptively fires off a localized burst of custom-made painkillers to brace for impact.
It's a neurological airbag deploying just as the crash occurs.
That's incredible, but observation isn't enough in neuroscience, right?
Yeah.
You had to prove causation.
Always. They had to prove that if they blocked those specific natural opioids, the placebo effect would fail.
Now, normally, you'd just push a tiny metal tube cannula into the brain and inject a drug blocker.
But you can't really do that here. The brainstem is densely packed and incredibly delicate. Pushing a metal cannula into the vlPAG causes physical tissue damage.
And that damage triggers neuroinflammation.
Exactly, which profoundly alters the very pain circuits the researchers are trying to study. It's like trying to fix a Swiss watch with a sledgehammer. The tool destroys the environment.
So they needed a drug that could get into the brainstem without causing damage. And they solved this using a smart bomb approach with a light-activated drug cage called PHNX.
Right, photoactivatable naloxone. Naloxone is a powerful blocker of opioid receptors. But they didn't just inject raw naloxone.
They used a specially engineered version where the active drug is trapped inside a molecular cage.
And in this caged state, it is completely biologically inert. You can inject it, and it flows harmlessly through the brain tissue without interacting with a single cell or causing any inflammation. It does absolutely nothing.
Until you expose it to a very specific wavelength of light. The researchers used a non-damaging, hair-thin optical fiber to shine a precise flash of light directly into the vlPAG.
The light hits the molecule, the cage shatters, and instantly, a highly localized dose of naloxone is released exactly where it's needed and nowhere else.
And when they flashed that light and uncaged the blocker right as the mouse was placed on the hot plate,
the placebo effect collapsed.
wow.
The endogenous opioids were blocked from binding to the receptors. This proved definitively that the cortical CEO can yell down the nerve fibers all at once, but if the brainstem cannot access its own internal pharmacy to produce those chemical keys, the volume knob cannot be turned.
The pain remains entirely real.
Exactly.
Okay, recap time. Bullet 1, placebos trigger a localized release of your body's own natural opioids right when pain hits.
Yep.
Bullet 2, cutting-edge light-activated drugs prove these opioids are strictly necessary in the vlPAG for the placebo to work.
Nailed it.
So listener, your brain manufactures its own custom painkillers. What other internal pharmacies might we be completely unaware of?
That is a fascinating thing to think about.
It really is. Okay, so they proved this specific circuit works for a hot plate. But life isn't just hot plates. Does this placebo shield protect against other types of pain?
And that leads to what I consider the most structurally significant finding in the entire paper. Placebo analgesia generalizes.
Generalizes.
How? To test this, they took mice that had been successfully conditioned to expect safety from the thermal pain of the hot plate. But on the test day, they completely changed the threat.
Instead of heat, what did they do?
They subjected the mice to mechanical pain, a sharp, uncomfortable pinprick to the paw.
Wait, that's a completely different sensory pathway, right? Processed by different peripheral nerves.
Completely different. And yet the placebo effect transferred perfectly. The mice that expected relief from the heat also demonstrated profound pain suppression when subjected to the pinprick.
That blows my mind. The expectation of safety acts like a universal shield, not just a specific localized patch.
Exactly. It created a generalized, holistic state of physical resilience. And the clinical implication for that is staggering. Because the researchers pushed it even further.
What do they do next?
They took completely healthy, uninjured mice and put them through this preventive placebo conditioning. They basically taught their healthy brains how to activate this generalized shield.
Okay.
Then, weeks later, they subjected those mice to a simulated chronic pain condition, a state called allodynia.
Let's define allodynia real quick. That's a neuropathic condition where the nervous system becomes hypersensitized.
Right. Normally, harmless sensations like a light breeze or a gentle touch are suddenly perceived by the brain as excruciating pain.
It's a hallmark of many chronic pain disorders in humans.
Yes. But what they found was that the mice who had received the preventive placebo training in their healthy state showed massive, lasting resilience to the allodynia.
Even after the injury.
Even after the physical nerve pathology was introduced, their brains aggressively utilized that pre-trained top-down circuitry. It suppressed the chronic pain.
They had literally built a fortress of neurological armor before the invasion even happened.
The translation potential here is just enormous. If we can condition a healthy mammalian brain to build resilience to future unforeseen pain, it fundamentally shifts how we might approach human medicine.
Imagine a future where, in the weeks before you undergo a major invasive surgery, your doctor prescribes a course of placebo training.
Resilience conditioning, exactly. You actively train your prefrontal cortex to establish these inhibitory pathways so that when the postoperative pain hits, your brainstem is already primed to aggressively mute it.
Final recap for this section. Bullet 1. The placebo effect isn't picky. It generalizes across entirely different types of physical pain.
Yes.
And bullet 2, preventive conditioning in a healthy state can build a neurological armor against future injuries. So, listener, if we can train the brain to be resilient to future pain, could doctors one day prescribe placebo training before major surgery?
I think we're heading exactly in that direction. It bridges the gap between psychology and physical medicine in a way that is just undeniable.
So what does this all mean? When we started, we talked about how the medical field used to view the placebo effect as this murky, imaginary nuisance. But Lovrici and this team have illuminated the exact blueprints.
They really have.
A learned expectation activates the executive cortex. The cortex sends an electrical command down the neurohighway to the brainstem. The brainstem releases a perfectly timed burst of natural opioids, which physically twist the volume knob on the spinal cord to stop pain signals from entering consciousness.
It's a proven, tangible machine. It is a masterclass in modern neuroscience.
It really is.
And I think it leaves us with a profoundly unsettling yet fascinating philosophical reality to consider. We've spent this entire time proving that your cognitive expectation can fundamentally override and alter the physical reality of pain via a dedicated neural highway.
Right. It aggressively edits the incoming data based on what you believe.
Yes, precisely. But pain is just one stream of sensory data. Our brains are constantly processing temperature, sight, sound, fatigue, digestion. If our nervous system comes pre-installed with this incredibly powerful top-down hardware designed to mutate our physical reality based purely on what we expect to experience, what other aspects of your daily physical reality are actually just hallucinations built by your expectations?
Wow. If my belief can physically turn off a hot stove, what else is my brain quietly editing right now just because it expects to?
It's a wild thought.
That is a thought that is going to stick with me for a long time. Thank you, learner, for joining us on this deep dive. Keep questioning your reality, and we'll catch you next time.